Welcome friends to my YouTube channel Bioscience Patshala. In this video we are going to discuss about the plasma membrane proteins. Plasma membrane proteins are larger in size than lipids. They are classified into three types. First one is extrinsic proteins, peripheral membrane protein. Second one is intrinsic proteins or integral membrane protein or transmembrane protein. Third one is lipid anchored proteins. So in this diagram you can see this is the plasma membrane, this is the lipid bilayer, and this is non-polar tail and this is the polar head. The extrinsic polar head and also it attached to the intrinsic protein, whereas intrinsic protein is present in the periphery and the lipid anchored protein is attached to the lipid bilayer. First one is extrinsic protein or peripheral membrane protein. Extrinsic proteins are present in the exoplasmic as well as cytosolic phase of plasma membrane. They are joined with the membrane lipids or membrane protein with ionic interaction or electrostatic interaction. The bond present between the lipids and proteins are ionic bonds or electrostatic bond. To isolate peripheral membrane protein, we use high pH or salt treatment. For example, spectrin, aniserin, actin. These are all present in cytoplasmic phase of RBC's plasma membrane. We take RBC as a model cell. To isolate peripheral membrane protein on cytosolic side, that is spectrin, we take a beaker in which we place RBC cell, and in beaker we pour pure distilled water. Due to the osmosis, the water by entering inside the cell, it swelled up and get burst or lysis. Then it turn into fragments, and these fragments are attached either inside out vesicle or right side out vesicle. Inside out vesicle protein are present on the outside, whereas right side out vesicle proteins, peripheral proteins are present in the inner side. So for the separation of inside out vesicle and right side out vesicle, we use affinity chromatography, in which we take bead and there is metallic arm attached to the bead and the ligand. The ligand we take is lectin. These have a high affinity towards carbohydrates. Then we take ligands in this flask. Then we see inside out protein is go down, whereas right side out protein, because of the outside carbohydrate, they attached with the ligand, and we separate out inside out vesicle cell. And then in this beaker we take we use salt treatment to isolate inside out protein, peripheral protein. Then we see integral membrane proteins. These proteins are spans plasma membrane at least once. Spans means they enter in the plasma membrane. The major interaction is hydrophobic interaction which stabilizes integral membrane proteins. Examples of integral protein is RBC. In RBC is glycophorin. The glycophorin is glycosylated protein of 131 amino acids. It spans plasma membrane one time. You can see this is we consider it as a plasma membrane where protein is present, spans one time, where amino end is present and carboxyl terminal end is present. Second example is band 3 or anionic exchanger protein. It spans plasma membrane 14 times. This protein made up of 929 amino acids. Third one is GPCR, G protein coupled receptor. It spans plasma membrane 7 times. Then we see the isolation of integral membrane proteins. We use detergents like SDS, CTAP. These are anionic charge detergents. These are amphipathic molecules, means they have polar head and non-polar tail. So in this diagram you can see this is the integral membrane protein along with the association of the lipid bilayer of plasma membrane. We use detergent, then this detergent dissociates the bond between the lipid bilayer and the integral membrane protein, but the protein we found or protein we get is denatured protein. For natural protein we have to use detergent like Triton Extend, which is isolated in native form. It is the no charge protein. The third protein we see is lipid anchored proteins. These are covalently bound proteins. They are covalently bound with either lipids or fatty acids. They are classified into four types. First one, myristyl anchored amide linked protein, acyl anchor thioester linked protein, prenyl anchor thioether linked proteins, and glycosyl phosphatidyl inositol anchored protein, in which first three proteins are cytosolic proteins, whereas the fourth one, which is glycosyl phosphatidyl inositol anchored protein, is secretory protein. So we see the difference between the cytosolic protein and the secretory protein. Cytosolic protein synthesis start in the cytosol on free ribosome, also in cytosol. They form in a cytosol and ends with the cytosol, whereas in secretory protein synthesis start in cytosol but ends in rough endoplasmic reticulum. The first type is myristyl anchored amide linked proteins. It is 14 carbon fatty acid. Myristyl is 14 carbon fatty acid chain. It attached with the amide bond with the protein. As in diagram you can see, this is the protein which has carboxyl terminal and amino terminal, and this is myristyl chain which has COH bond on the end, which is acidic bond. Then we can see the CO minus terminal attached with the nitrogen amino terminal and form hydrophobic myristyl chain. So these are located inside the plasma membrane. The myristyl chain is in the plasma membrane because the inner surface of the non-polar tail surface of the plasma membrane is hydrophobic, so it got attached. The example of myristyl anchored amide linked protein is G alpha protein. Second one is thioester linked acyl protein. It is acyl is the 16 carbon fatty acid chain. As we see, this is the protein which has carboxyl terminal and then amino terminal, and this is acyl chain which has COH bond at the end. Then it forms sulfate carbon bond or thioester bond, and on the CO minus terminal end, and NH3 plus terminal is it free. It also attached with the plasma membrane. The tail the acyl chain is attached inside the plasma membrane, whereas the other side is present in the cytosol. The proteinic side is a transferrin receptor and GPCR repair. Type of protein is thioethyl linked prenyl anchored protein. Prenyl is a terpene of 15 carbon. This is terpene and this is the protein. It forms sulfate terpene bond and the amino end. It is also present inside the plasma membrane. The terpene chain is present in the plasma membrane. Last one is GPI anchored protein. In this diagram you can see, this is the endoplasmic reticulum and this is the inside of the endoplasmic reticulum, that is ER membrane. And inside this we can see the protein and which attached to the phosphatyl bond which attached to ethylamide, and ethylamide is attached to the 3 or 4 sugars, and this is phosphatidyl inositol. Then phosphatidyl inositol bound with the sugars which attached with the ethylamide which attached to the phosphate bond and the protein, and this form the vesicle, and this vesicle is then integrated into the plasma membrane. Thank you.